All right. Good morning, everyone. Thank you. We are so delighted that you are here with us today. We want to thank you for joining us and welcome you to our annual research day and to what is now our 29th annual Bullough lecture and our 13th annual research day. I also just want to make a few comments and tell you about, we are so fortunate to have this endowed lecture in honor of Dr. Bonnie Bullough, who was a former dean of the UB School of Nursing from 1980 to 1991. And this fund was established by her husband, Dr. Vern Bullough, and she herself was an expert in human sexuality, and she published extensively over 120 articles and 24 books on this topic. She was both a family nurse practitioner as well as a pediatric nurse practitioner. She was also a fellow of the American Academy of Nursing, and then she joined UB after having held positions at UCLA and California State Long Beach. She had originally received actually her PhD and master's degrees in sociology, and she held a master's degree from UCLA and did her bachelor's degree actually at Youngstown University in Ohio. So we are so thankful that her husband set up this endowed lecture for us to make this an even more special event for the School of Nursing. Today, I can't wait to preview the posters and look at some of the work that's going on here. I know it's really exciting. I especially want to thank our community partners for being here because I always enjoy seeing what you are doing as one of our community partners here in the School of Nursing. I also want to thank all of our student presenters that are here today as well. I know that all that we do has such, hopefully, innovation, and the impact that we do to help make things better for the patients that we take care of is where we are really all targeting and focusing our efforts. I also want to acknowledge our Associate Dean of Research, Dr. Yuping Chang, and her office, Courtney and everyone in her office that worked so hard to make this event such a successful day for us. I also want to just briefly tell you that I am so delighted that we have Dr. Wendy Henderson here today. Dr. Carliera Weiss will be giving her formal introduction, but having both us, because we have both been at National Institutes of Health, we kind of carry a special bond. And I think we have this ideal thing that we always strive for is also trying to put the health of the nation in all that we do and reflect that mission. And you become a part of that, having been at NIH in a way that just helps to anchor, I think, your career in a way that I've not felt anchored in any other place I've been. But NIH did that for me. And so it's always special for me to have somebody who has been on that path along where I have been as well. So I first want to introduce our Carliera, who will be making our introduction to today's speaker. Dr. Weiss is an assistant professor here in the UB School of Nursing. She started our first basic science laboratory here in the School of Nursing, which is, to me, a milestone to help expand our efforts, where we can also look and begin to measure biomarkers and other basic science approaches to what we do. So again, if you need a collaborator who can measure some biomarkers, I would urge you to reach out to her. So this really helps to expand our ability to engage in some molecular and cell biology, biochemistry, and really look at what is happening in a biological way, not just in some of the behavioral measures that people often use. Her laboratory is specifically focused on looking at Alzheimer's disease and risk factors and sleep, and how sleep and cognition and Alzheimer's disease all tie together. She's been working internationally in this field, and in case if you pay attention to UB Now, you know she's a commonly interviewed across the news media, especially around every year when we change our light cycle and move from Eastern time to Standard Time, Daylight Time to Standard Time. So she's commonly in the news with that. So again, I will invite Dr. Weiss to come introduce today's speaker.
Thank you, Jean Masaki, for the introduction. We are honored to welcome Dr. Wendy Henderson as our 29th annual Bullough lecturer. I apologize for my accent. Every time I get a little too excited or anxious, my accent comes out. So I will speak slowly. Dr. Henderson is the Gail and Ralph Reynolds President's Distinguished Professor at the University of Pennsylvania School of Nursing and a Professor of Medicine in the Division of Gastroenterology and Hepatology at the Pullman School of Medicine at the University of Pennsylvania. As a primary investigator on multiple studies, Dr. Henderson studies the brain, gut, liver microbiota axis and chronic effects of stress on intestinal health across lifespan in human, murine, and cellular models. She co-developed the gastrointestinal pain pointer technology to provide clinicians with a more integrated tool to assess GI symptoms, one that includes location, intensity, quality, and physiological parameters. Through a brain-gut natural history study, Dr. Henderson team also evaluated the brain, gut, and liver interactions in persons with chronic abdominal pain of unknown origin. The Henderson lab at UPenn focused on developing disruptive technologies that are intended to be personalized, direct to customers, inexpensive, and impactful in resource-limited settings. Dr. Henderson is a fellow of the American Academy of Nursing and a senior fellow of the Leonard Davis Institute for Health Economics at University of Pennsylvania. In addition to her prolific research and teaching experience, her clinical career has numerous appointments as nurse practitioner in the Pittsburgh area. From 2008 to 2019, she also served as clinical consulting and nurse practitioner of pediatric gastroenterology, hepatology, and nutrition for the consulting team at the NIH Clinical Center. Please welcoming Dr. Henderson.
Good morning. So I wanted to start by thank you. I mean, this is a full house. Oh my golly, look at this. It's wonderful. But it's really a pleasure to be here. I just wanted to start with, I'm a nurse. Every question and everything I'll talk to you about today is not my own. They're not the questions that I'm asking. These are the questions or the persons that had problems that I couldn't help. And I brought together teams of individuals where we could find a way to give voice to persons in need. So I wanted to start by knowing my audience. How many of you are looking forward to May 15th? Student, parent, faculty. Okay. How many are already nurses? How many are looking forward to taking that NCLEX? Okay, not look at how many are going to be taking the NCLEX in the very near future. Okay. All right. So now I have a better idea of who we are here. How many of you really care about nurses and nursing? All right, we're all here together. So thank you. Thank you for joining me. And so what I want to talk to you about today is really a story of discovery. It's a road, it's a bit of a weaving road of discovery and a path, and just wanted to encourage you for how to show the value of nursing, where you're going, what you will be doing, and who you need to kind of bring along the way so that you can make a difference. Hang on. Did work. And now let's see. Yes. All right. So here we are. So of course I didn't make this picture. One of my PhD students in the lab used AI, and I was like, how can we have Ben Franklin walking, and so this ounce of prevention is worth a pound of cure. So I'm also a pediatric nurse practitioner in primary care, and many times along the way, regardless of the study or the cohort or the patient, I kept thinking, geez, if we could have just done something sooner, right? If we could have, would have, should have. And how can we as nurses be a part of that path? And how can we think about this ounce of prevention and cure? So again, I'll take you down this road of discovery, but it really started with, now who knows what year, Hemocult. Does anyone even know what this is? This is a Hemocult card. How many people know what a Hemocult card is? Okay. The other people are like, what in the world is that thing? All right. So back in the 90s, in clinical practice, we would actually keep a whole stack of these cards in our pocket. And then we'd have this little vial, which was reagent, and we could put sputum, stool, any kind of liquid human product onto a little bit on the card, flip it over, put a dropper of this reagent, and it would light up. And if it lit up blue, then we knew that there was blood present, right? About this same time, the thinking was that, well, you could be colorblind, which is true. The reagent could expire and it could be poisonous. And here you are in a pediatric primary care setting, right? You shouldn't be carrying this around in your pocket. But what did I know? You know, I had treatment algorithms. I knew if there was blood or hematochezia present, I knew what to do. And suddenly, these cards weren't allowed to be in our pockets anymore. And this haunted me because where I was at the time was Children's Hospital of Pittsburgh. I was in rural clinics in rural Appalachia. And this really helped me. I was new. I was novice, right? 1999, brand new nurse practitioner. You know, how many years ago was that? Oh my golly. And geez, it would keep me awake at night. Babies coming, breastfeeding, babies with blood in their stool, maybe not. It's diarrhea. How do I know? Do we change the formula? Do we change the diet? You know, is this infectious? So as you practice and where you go and what keeps you awake, because you're not sure if what your choices and what you're doing is the right thing or the standard of care or the orders that you're following, you know, be thoughtful. Think about these questions, these problems that you have. You'll have a very different problem, you know, in your world as it comes forward in the future. But what will that be? And how can you keep this in your pocket, so to speak, so you can keep thinking about how you can make a difference? So what did I do? Well, I did the best I could. So I'd say, okay, call me back in a week. Let's see if we try this. Call me back, let's do this. And then before I knew it, they couldn't call me unless it was billable, right? It had to be a billable service. We didn't have telehealth. We didn't have these kinds of things. I couldn't send them home with a stack of cards that they'd send back. And then I could tell them, okay, that made a difference. Now we're not going to do antibiotics or we're not going to do this, et cetera, et cetera. So thinking about this road took all the family's resources to bring these children to our clinic, and they're seeing me, right? So you want to give as much as you possibly can. What did I wind up doing? Worked for 5 years as a nurse practitioner, involved in research, involved in training the fellows and so forth, loved my job. And I just kept being challenged with, gosh, see more, do more, see more, do more, right? So 15 patients a day, come back, be on call to the emergency room, go to McGee Hospital, children with apnea bradycardic episodes, go to the, right, go. And I thought, wow, okay. So in about that time, the Children's Hospital was merging with the practice plan, University of Pittsburgh Medical Center. They said, okay, you're a nurse practitioner. You can do flex SIGs. You can look for rejection in the biopsies. You can see constipation, reflux, Crohn's. Now you can do anything, hepatitis, name it. If there's an algorithm to follow, correct? And I thought, do I want to keep doing what I'm doing? Because they'd move all nurse practitioners to the practice plan because we could bill and see more and dictate and so forth, right? And be on call. Or did I want to keep moving towards how could I make a difference, right? How could I give back to the communities in need? And so I went back for my PhD. I had to make a choice, right? They say, oh, we'll pay you more. You can, we'll get longer hours. What do you want? What do you need? And that wasn't what I really wanted or needed. I really wanted to make a difference. And I was still awake at night. Did I make the right decision? Was that the right choice? So I went back to school, went back for my PhD, wrote something called an F31, which is a pre-doctoral grant that's sort of a golden ticket that'll fund you for your PhD. I was like, I can do this. I've got the best question, right? This is exactly, back then there were pink sheets. This is exactly what it said. There is no evidence that your problem exists. They were right. There was no evidence that me not having something in my pocket or the numbers of patients that I was seeing, there was no evidence. I had written no paper. There was no evidence of what I knew and what I knew, the algorithm or how it could change. They were right. There was no evidence. And I was like, what are you kidding me? What? I can get the data. No. Think about this. What evidence do you need for the problem that you care most about? And how will you demonstrate that? And there's so many new ways to get this evidence, right? Finished my PhD. Didn't get that F31, right? Couldn't do the PhD in pediatrics the way I wanted to do because there was no evidence. But I learned the process. I learned how to do research. I had wonderful mentors, Judy Erlin, Corinne Barnes. And I did a first postdoc in big data. This was the big data. We'll talk a little bit about administrations. We are being recorded, so I'll be thoughtful in what I say. Although I am no longer a federal employee. I did a first postdoc after my PhD in big data. So Harold Varmus was at the NIH, and this was the big data. You're like, what year was that? That's 2007. We're back to big data again, right? It comes full circle, just like your heels and your pants and your outfits. So I was recruited for a second postdoc then at the NIH. I was coming there with this, how to do structural equation modeling and big data, AI. We didn't have that word yet. And I got there, and guess what? The administration changed. And there was a new appointee. So who came next? Who knows their history? Who was the next head of the NIH? I'll give you a hint, human genome. Francis Collins. So Francis Collins comes in and says, okay, all postdocs, anyone funded has to have genetics as a part of their science. I did not have genetics as part of my curriculum. In nursing, I didn't know anything, right? Other than what I'd learned in GI. I knew OMC, this, that, Pianka. I knew about, different treatments for colitis and so forth that had genetic modified. But I had to learn genetics. This is how you pivot. Where are we now? How are we changing what we want to do and how we want to do it? And what do we might or need to incorporate, and how could it be relevant to all? So I did it. 2 years at the bench learning immunogenetics. What did I do? Luckily, I still had collaborations back from Pittsburgh from the clinics. There were many participants that had agreed to be contacted. We brought in biopsies, pre-existing biopsies, so there was no added risk. And I focused on other patients also that I couldn't explain how I was helping. So the other big part of my job wound up being patients that had what was called NPD, no pathologic diagnosis. They'd had an upper and lower endoscopy, right? So endoscopy, colonoscopy, biopsies, had chronic symptoms, but there was no evidence their problem existed. I've heard that before, right? I thought, I'm going to get some evidence. Let's get some evidence, right? And if we've got to learn genetics and find evidence, we can do that too. But how can we do it without doing harm? And a very small budget, because I was in the Nursing Institute, right, as a postdoc. So they said, oh, you're fully funded. Well, that was my salary. There was nothing else that came with that funding. Anyway, so this is the National Institute of Health. This is the clinical center. Let's see if the pointer kind of works. Let me find the mouse just so I give you perspective. So this is, although I think in Dubai there might be a larger brick and mortar research facility now, but over 4 million square feet, pretty much the largest research facility, definitely in the United States and most of the world. And it's a hospital. So there's no emergency room, there's no orthopedics. Every patient that comes is on a protocol. They sign an informed consent. There's no fee for service. There's no see more, do more in the clinical center of the NIH. Patients can come from all over the world. They can be housed and they're paid and transported to come to receive care. So I thought, okay, I'm here. I've got to learn the bench on genetics, but I still want to be a practicing nurse practitioner. Is there any way that I can, help in oh, sorry, no, postdocs aren't allowed to do that. You're not covered under the federal torts law. You can't do that, can't do that. So, well, you could be a staff scientist, and as a staff scientist, technically you're an employee. And I'm like, oh, does that mean I get paid more than a postdoc? Maybe, yes. And I could pro bono see patients as a nurse practitioner too. So that's what I did on my extra time. There were only 2 of us, an attending and me, for all pediatric patients coming in and with GI problems into the clinical center of NIH. So it was an amazing experience for 10 years. But what was I always kind of thinking about? What are these problems? What is non-pathologic diagnosis? What are the symptoms that people have? And me, pediatric gastroenterology. What is gastroenterology? What are the biggest symptoms that we have? reflux, abdominal pain, diarrhea, constipation, right? So the National Institute of Nursing Research is a disease agnostic entity. So if it's cancer, your funding is in the Cancer Institute, right? If it's infectious disease, you should be in the infectious, NIAID in terms of infectious disease. So for me, it's like, well, okay, how do I fit here? I've got to do genetics. I've got to do, right? We can do this, right? Nurses, we can do anything. How do we pivot and work on this? And how do we imagine a theoretical model, which is what we do? So we developed something called the symptom science model based on symptoms that our patients tell us, wherein we'd need to phenotype or have phenotypic characterization, biomarker discovery, clinical application, and on around, right? And then for me, how could I apply this to the patients that I cared about, right? And that were in need. And what about all the patients that I would see because there was no pathologic diagnosis that had history of chronic abdominal pain? May have had alternating bowel habits, constipation, otherwise. This sounds like a symptom-based phenotype. This is before the year of calling something called irritable bowel syndrome, right? And of course, if you're in this precious real estate where there's no fee for service, it has to be something where no one else could do it. So what did I call it? I made it up. Chronic abdominal pain of unknown origin. It was chronic recurring abdominal pain, but that was crap, and they wouldn't let me get paid to do crap. So anyway. So at any rate, this was the model. This is how we operationalized our model. This is how we justified what we were doing. And this is how I thought, you know what? Still got that thing missing in my pocket. I'm still thinking about that. But let's stay focused and continue the training. What I'm going to walk through today, and we'll do it somewhat quickly, but you can feel free to contact me or look up any of the papers. This is 10 years worth of work, is to walk you through how we were able to develop a better way to phenotype person's symptoms, how we could recapitulate their symptoms in real time, as well as to have a biomarker discovery that could be direct to the person for self-care, and then how that kind of feeds that cycle of how it could improve and know how to treat and move forward in terms of maybe preventing their symptoms. So key facts about digestive symptoms: up to 20% of the US population report digestive or GI symptoms, 30 billion in costs annually. Abdominal pain, the most common GI diagnosis, single most common reason for ER visits, top 10 for outpatients. We all know this. Doesn't matter what you study, persons have GI symptoms, right? It's many and varied. What did I need to do? I needed to show that persons that I saw, and I couldn't explain what I knew or how I knew how to treat kids that would come with chronic recurrent abdominal pain. Okay, I won't call it crap anymore. But how would we know? We talk about, people would say, well, it's all in your head. You just need to relax and not be stressed, and then you won't have symptoms. This is what they heard. They'd say, why don't you go see, we have an abdominal pain nurse practitioner. She has a clinic in, these outpatient clinics. You can go see her and see if she can help you out. And we do journals and diaries, and we keep track of these symptoms, and we talk about it, and we do stool diaries. And I was like, there's no ulterior gain. Like, it's summer. They want to go to that birthday party. They don't want to miss it, right? And there must be something here. And after a few years, you'd start to say, hey, I wonder if I try this, if this would work. Or I wonder if I try that. But then I couldn't understand, how did I know that, right? And as nurses, and you graduating in the next generation, regardless of your program, how will you teach that next generation what you know? Where is the evidence that that problem exists? And if they say it and if they feel it, isn't there some way we could quantify it? There must be a way. So let's talk through this. So again, all those biopsies coming in, looking for genetics, learning to slice and dice, use a microtome, right? The problem that we had was, how do we ask people about how they feel? Okay, we've got lots of anesthesia folks, and how do you ask about your pain? What's your pain? Okay, on a scale of, 0 to 10, 0 being no pain, 10 being the worst pain ever. Or kids, what do? Maybe we had some faces, like, how do you feel? Show me how you feel, right? So what's the problem with quantitating that way? Right? How do you detect a change? And when Joint Commission comes in and says, for payment for performance, you need to detect at least a 30% change. And you do an intervention, your patient comes back and they're like, well, what did I say last time to the parent? Or, oh, was it this or did I pick that face? And is that face to this face really a 30 to 20% or enough difference for a clinically meaningful change? How will you show this? So we devised this GI pain pointer. This went through so many renditions. Pilots, I had like the esophagus was red and the stomach, you could see it, and then they'd be like, oh, where's my stomach? Tanner staging not being shown here for obvious reasons. Gender, you can pick your gender, you can tattoo, you can put hair, you can pick your skin color. And so this is on an ultra mobile PC, but what do you not see? You don't see a 0 to 10, right? We also did a different facial morphing technology thing for the Whaley and Wong kind of thing too. But anyway, so what participants could do, they could pick their body type, they could dial up how much it hurts, and behind the scenes, there'd be a 0 to 100, click, 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 and then the thermostat goes up, and this is like the McGill pain scale in terms of descriptors. We'd add descriptors if there were words that they'd use that weren't words that were on the tool because it wasn't relevant for their culture or who they were. Normal weight, overweight, they can morph their body. It can flip over. You can have multiple pains. It can be a headache. It can be tenesmus. It can be abdominal pain, etc. And it would capture how big the pain area was via binary and executable code in the background in real time. And then it would sync with a Bluetooth kind of device that would capture blood pressure and heart rate. And then during COVID, we also added pulse ox. So all that data would capture in real time. And lo and behold, because it was so simple, the technology, it could actually work via telemedicine and capture to the provider's site de-identified. Also 508 compliant and so forth. This is simple, but why did we need this? We needed to be able to show what we did as nurses and what we did for our interventions, that it worked, that it made a difference, and show that value of what we did. So this is being used. It's freely available if you're interested. It belongs to the people of the United States through the federal government. You just have to ask. And we validated this and published it. So back to those biopsies. No evidence a problem exists, right? No pathologic diagnosis. So I thought, geez, if we could standardize their pain phenotype so we know exactly how much pain they had because they told us. And then if we could do some sort of comparison and look at markers for pain to show that there is some physiologic marker of pain for patients who were told it's all in your head and there's nothing wrong, right? And could we also then show maybe why some of the things we did worked? And we didn't know really why they worked, but there might be evidence. So we stained for things like interchromaffin cells, 5-HT. Who knows the other name for 5-HT? Serotonin, 95% found in the gut, only 5% in the brain. I know. I'm gut biased. Substance P, a pain marker. Why in the world would we find increased substance P in the lamina propria if there's no evidence that you have anything wrong with you, but you have pain markers in your gut? Interesting. So this was enough evidence. Of course, I had to learn how to deal with this at Fort Detrick and animal labs and so forth. But we found a way. And how could it be? How could it be that if there's no pathologic diagnosis, you have an increased number of mast cells compared to an inflammatory phenotype in normals? How could that be? There must be something more. So these little bits of evidence allowed me to continue on with the team and ask other questions. And of course, I think simply, how does this work? And there's all these pain pathways for the pain experts. Some of you may be in the room. But I'm like, but how could this be? There must be some memory. The minute someone thinks about something, they may have a recapitulation without even an intervention. What is that memory? How does it work? So I was very curious. And how in the world could that substance P be in the lamina propria, and how did it get there? And how does it get through? It's just simple science. And how can we show this evidence? So about this time, I had finished my postdoc. I was now something called an assistant clinical investigator, where you get a little bit of money. I think I had $15,000 a year for the first year, which was a lot, considering. But I was actually allowed to bring participants in. I could bring participants in who had nothing wrong with them, chronic recurrent abdominal pain. So we called it abdominal pain of unknown origin. And to carefully phenotype persons by what? other inflammatory states, right? To be really thoughtful, right? That's sort of where hindsight can kind of be. You've got to really be thinking about how you provide that evidence where someone wouldn't say, oh, but that could just be inflammation because of X, Y, or Z. So to really be very careful in terms of our phenotyping, including ovulation, bringing in female individuals during the luteal phase, right? Because middle submerge, you can have abdominal pain related to that. Like think of any other reason that people say, oh, no, there's no evidence, or oh, this is flawed, right? Being really thoughtful. And so we did indeed find with the hypothesis there was an underlying inflammatory mechanism related to chronic abdominal pain of unknown origin. We found indeed that there was CCL16, which is a liver-based chemokine. Liver. Okay, I'm in GI liver. What is this? What is happening? So again, somewhat biased because it's a predetermined inflammatory array, right? So there's bias here. It's not shotgun sequencing or anything really sophisticated like that. And then along came these amazing things that now you had, but I didn't have, where you can just click on one of these lines and like 10 papers show up to tell you that relationship between these two molecules. I'm like, what is this? I don't understand. But you can very easily. And then I was like, well, how does this relate to antibiotics, to inflammation, to pain? And then you can further interrogate. This is not rocket science. This is something that's really kind of simple if you just think about it. And you can plug and play and find out more about your area of interest. I still wanted to know what is small enough to permeate the blood-brain barrier and get through the gut. How is this happening? And so microRNAs are that small. They're small enough to be held in little tiny purses called extracellular vesicles that permeate the gut and the blood-brain barrier. And then I was like, I wonder if there's a way that we could look at this in real time, where if I'm out in the community somewhere, if I could phenotype and know someone's diagnosis and know what to treat, even if they couldn't necessarily tell me, right? Babies can't talk to you and tell you exactly, right? So at any rate, so we were able to look at microRNAs. We were able to look at the full gamut. There's a patent. It's publicly available. You can look these things up online. If you're interested, belongs to the U.S. Public. And we could tell from, and this is pre all the, I don't even know, I won't say the names of any persons that got into trouble for drop of blood kinds of things, but this was before all of that, where we could tell basically from a blood draw if someone had irritable bowel with or without diarrhea just from their blood. So back to the symptom science model, still getting to this phenotypic characterization, and I'm thinking, okay, liver-based chemokine. And I'm thinking endotoxin, right? How do things get through? And where is there, right? The quiver in the liver. And I mean, just simple thinking, right? Here I am, pediatrics, like I don't know all the details, but trying to find out, how am I going to prove this? How am I going to recapitulate patients' symptoms in real time without harming them, right? There's lots of ways you can recapitulate something. I didn't want to harm anyone. And so what we did, and this was with help from someone named Rob Schulman at Baylor, a pediatric gastroenterologist, shared a formulation of a way to look at leaky gut, right? To look at gut permeability. And he had a combination of sugars with some other materials that in kids, we didn't really know if it would get in or out of the brain. And so I was like, could we just do this with simple sugars? And so what we developed was something called the brain gut test solution. It's simple. And we'll see how many of you know what some of these things are. Okay, so it's a combination of lactulose. How many of you know lactulose? Yes. What do you use it for? What else? Who are the ICU people in the house? Ammonia for what? Yep. Do we know how it works? Not really. We know that it's osmotic laxative. It pulls water into the gut, right? It decreases levels. It works for hepatic encephalopathy as well. We don't fully understand how. Now we better understand it's the microbiome that breaks down anyway. I'll give some of that away. But so basically it's sucrose, sucralose, lactulose, and mannitol. So these were 4 different sugars. They had different molar mass. What does that mean? It's just the size of the sugar particle is different. And so it can permeate the gut at different points. So then I can tell when they drink it and when in a fasting state and you capture what's excreted in the urine, potentially where that problem is, where they're having permeation of the gut from the brain gut test solution. And we did this. What we found, which was unusual, was that persons who had a history of chronic abdominal pain, placebo or not, once they got this test solution of sugars, they were 20 times more likely to get abdominal pain with the test solution than someone without. We didn't even need to do the mass spec or collect the urine. We knew within 15 minutes if they got abdominal pain with the real sugars. We knew where it was and where the problem was and potentially what we may need to do to treat. So, and then this gets back to the whole liver in terms of like positive, negative controls. And I don't want to bore you too much, but again, they'd say, well, where's the evidence? Like, oh, they're saying this, but where's the evidence? Like, do you have some blood marker? Do you have this? Do you have that? And so indeed, we could look specifically at soluble CD14 as well as LPS, but instead of LPS, because of course you'd be criticized because it's acute phase reactant, it's endotoxin, blah, 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 it could be contamination. So we looked at LPS binding protein, which was from the liver, which those together are macrophage activation, and that could be that memory. Again, works in progress, early days. So this brain gut test solution, we've gone to different research pharmacies around the country, from Harvard to Rockefeller to the West Coast, and they're using this in different protocols, usually drug studies of gut-hone disorders. Who knows some gut-hone disorders? Name them. Come on, I got to keep you guys awake. HIV. What's another one? Cancer. Another gut-honed. Colitis. Mastocytosis. Keep going. Allergy. Name them, right? So at any rate, so we published our method in a chemistry journal, and they're like, what is a nurse publishing in a chemistry journal? You know what? We can do anything. So along came Polly Joseph. And Polly Joseph did a postdoc in my lab, and she was interested in taste receptors in the gut. She was like, hey, I heard you've got this sweet taste solution, this brain gut solution, and could I look at your data, and there's this new thing coming out, microbiome, could we look at some phyllo chips and see if there's some differences based on the sugars? And sure enough, we found it. What do you see on here? What's missing? Look at the lines. Where is there a missing line? Yes. What is the value of nothing? Well, it's something in the microbiome world, let me tell you. So the plus minus is whether or not they got pain from the test solution. The IVP is the induced visceral pain from the GI pain pointer. That's how much they dialed up on the pain pointer. So we'd give that, we'd ask their pain on the pain pointer. We'd give them this test solution in the clinical center in a fasting state. And then you can see that the excreted lactulose in the urine, what is it? Who knows data? It's highly correlated. So the more they excreted of lactulose, meaning the bigger the more gut permeability they had, the higher their pain. We kind of knew this because they were telling us this, but then we had the data that went along with it. And we found that, what if they give us a little bit more funding? So we look at something more than operational taxonomic units. This is kind of old school language for microbiome. If we looked at, I thought it was all in your head. Why in the world would you have mycoplasma? And why would that go with more pain with the test solution? This is very interesting. So this is more data, evidence the problem exists, evidence that it's not all in your head, and that your pain may be of unknown origin, but it's real. And it's related not only to your genetics, to your gut permeability, but also to microbes. Again, simple science. I'm like, oh, okay, I never had these courses either. I have a primary care pediatric nurse practitioner, and I have to learn. And I'm like, can you just show me where these particles are, cytokines, whatever. Where is the cell membrane? Where are they in the peripheral circulation? What is it, and how could it possibly get through the gut into the brain? Like, make this simple for me in a way that's understandable. And so what you see at the top here is this is the plasma membrane. And red means something is downregulated. This is an RNA perspective. So this is peripheral blood from the same patients, same one, same time. And so red means it's down. Green means it's up in terms of RNA expression in their blood. And again, I'm like, what am I going to do again, I've got to do genetics, right? Got to do genetics, but how's this going to help me in real time with my patients, right? So what was interesting was something called CD9 was down. CD9 is relevant because CD9 is a marker for extracellular vesicles. Extracellular vesicles permeate the blood-brain barrier. What else was down was something called lysozyme. What is lysozyme? Lysozyme is found in tears. It's found in breast milk. It's found in surfactant. It's found in the mucus lining of the gut. And it's down in patients who had pain. Why would it be down? So here I'm getting really curious. And of course I didn't even ask, were they breastfed? Of course I could go back and ask, this hindsight, was it used up? Were they born that way? right? Questions. And I'm going deeper, 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 deeper. So we had to learn electron microscopy, of course, we're nurses, right? Well, you could do anything. If you could put in an IV line, you can do this, right? 600 hours later, they finally let us actually handle jitterbug, the electron microscope. And of course, we had to be able to learn how to isolate extracellular vesicles. And the gold standard was, oh, you've got to tag them and pull them down with this magnetic. I'm like, well, but then would it still be viable? Could we humanize our animal models? And they're like, well, why would you want to do that? I'm like, because they're going to ask me, where's the evidence, right? How am I going to show this? Sarah Abbey is here. Sarah Abbey is now a nurse practitioner. She came, I found her pushing people around the NIH as a volunteer because she was disenfranchised being a cancer biologist. And she wanted to talk to patients. And I had no funding, so I kept going to the volunteer office saying, is there anyone that wants to help me in the lab that can, Anyway, before you knew it, our brains together, she knew what I didn't know. We found Joanna from the Netherlands, who was actually using something very similar to pantyhose to filter blood so that the extracellular vesicles stayed viable, right? So that we could then look at them under the microscope and tag them for exactly what we're looking for to show indeed, right? To show indeed. So what are these, some of this, I don't want to bore you, but we wanted to tag indeed that they were CD9 labeled, meaning that they were extracellular vesicles, not just how they looked, but proof. And then LBP was that LPS binding protein plus meant it was high in the blood. So again, this was biologic, not just what we knew they were 20 times more likely, right? Have abdominal pain. We knew this, but we wanted proof. And then this was mucin 1 and 2, that's gut markers. So where is it from? How can you show it really got through the gut, right? And then just normal control sort of things in males and females. You could actually watch the vesicles. You could watch them reach. See that horseshoe crab kind of thing, kind of reaching. Really interesting work. Now this has moved to cryo-EM and atomic force, and there's all kinds of other ways you could do much more with this. But you do this, and then you sit back and you're like, okay, so what? I've shown the problem exists. I've shown the molecules that permeate. I have shown that this is real with the team, not me alone, of course. Along comes Annette Wasaki's work. Dean Wasaki. I was like, oh, wait, there was someone in this lab. They have this equipment over here. There is some way that we could do this really in a way there's proof. Someone else did it. Let's just follow what they did. Call up Dean Wasaki. How do you do that essay? What do you do? Simple science, scratch wound essay, because I mean, it took us a while to learn to grow cells and, epithelial, colonic epithelial to show indeed that if we put lysozyme, this cocktail on these cell culture membranes, that it actually would heal better or worse, or what was it? And how could you show that? Keep the cells that survived the wounding. Who knows what any of these markers are right here? Who knows what PDL1 is? Who's heard of that before? Oh, get ready, cancer world. You're going to hear a lot of it in the next year once you're out there. New medications that are actually healing. They're also anti-cancer. So this pain that our patients were having may have actually been helping them, right? They've actually, their wounds heal faster. Their gut heals faster, right? When you put this cocktail of lysozyme, yes or no, and then you scratch the epithelial cells and cell culture and show indeed mechanistically that this is real. Back to the gut. So here's Nick Forey. Everyone was after the druggable target. I'm a nurse. I'm the forest, not the one tree. If it's just one tree, it can't possibly, what is this forest and what matters? So Nick was an evolutionary biologist, and he thought differently, the way that I was thinking holistically. And so when he came to the lab, we started to ask these questions, holistic questions of the microbiome, like not just what is or isn't present and what's up or down, but what is the milieu? And isn't there a way analytically, I did all this data science stuff, right? That we could look at the data as a whole. And so this is something called a Lefse analysis. It's kind of like a glorified t-test for any of you who've had statistics. But really what it showed, I wanted to know, what could I tell my patients? What do you eat? What do you do differently? And how could we relate this specifically to you from a personalized profile? And so we did indeed find that there were differences, and what this in terms of, and I'm very much oversimplify it, go and read the paper if you want to be able to, I can't even pronounce all the names of the microbiome, let alone spell them. But what it showed us that the short chain fatty acids that are related to the combination of the differential expression of the microbiome were related to butyric acid. Butyric acid and butyrate is found in carbohydrates. Carbohydrates. So they said, oh, this can't be real. You got to show us this in an animal model. I was like, I am not doing animal science. I am not interested in doing Baristat, colonic Baristat on rats and all these things that they wanted me to do no, thank you. However, there was another investigator, John Wiley at University of Michigan, who was like, oh, yeah, I'm not using those colons. You want the colons? I'm more interested in the brain. I said, oh, great. He had a model. It was a water avoidance model where he would chronically stress the animals. So basically, the chronic stress model, the animal goes on an island for 1 hour a day for 10 days, and they get stressed when the water comes up because they're afraid. So it recapitulates this sort of stress response. If you gave me 1 hour a day on an island without my children, my husband, okay, no, okay, never mind. But anyway, so, and we did indeed find similar findings in the stressed animal model versus the non-stressed healthy model, as we found in our human model. So, and this again, the left's the analysis, and it came back again to butyrate and the diet. So now we're like, oh, okay, let's go back to this cell culture model. Dean Wasaki, help me, what are we going to do? And I was like, okay, stress. So they say, oh, you're stressed, it's all in your head. So is there a way we could stress the cells, not stress the cells? There's lots of models of stress, whether it's metabolic, whether it's mechanistic, right? So you can think of stretch, there are different ways. But for me, I wanted somehow, so we used dexamethasone. Not perfect, but a model. And we first had to chronically grow the cells I didn't want antibiotics, right? And this is colonic epithelial cells, of course, the colon. I'm keeping you all awake with all my gut talk, right? So we chronically stressed the cells, kept them alive, and then we put dexamethasone on them, and we saw no difference. And what this is, transelectrical epithelial resistance. It just is cell permeability from a cell culture perspective. You basically take these two kind of chopsticks, stick it down in there, and it gives you these ohms that tells you the permeability. It's not complicated. It's really pretty simple if you learn how to do it. But again, we found no difference in permeability if you put the byproduct of the microbiome, which was the butyric acid, right, butyrate. We found no difference if you chronically stress them other than what you would normally find. It was the interaction. It was when they were stressed and the byproduct of the microbiome. So it's not just being stressed, but it's being stressed and what you eat. Think about that. So and then we had to learn confocal microscopy. We had to show indeed that, the tight junctions and the gut permeability, right, of the cells. So we can do that, right? And show occludin, which is a transmembranous protein, and ZO1 is a scaffolding protein. And then you just take pictures basically with a confocal microscope with and without the dexamethasone, with and without the butyrate or the byproduct of the microbiome. And you can see all those nice, pretty lines. So when it's untreated, they're nice. So these are the tight junctions. See how they're nice and straight. When you add the dex, they get a little fuzzy, and then they really start to break down with the merge, or when you put them both together. This is KEGG pathways. Now you could just ask AI probably to do all this for you, but we had to go through every little mechanism and plug it all in. And Jeff Robinson here was the computational biologist who helps with this. And this is what we did over time and what we showed. So this is, we're presenting this next week, actually at the diabetes, not the diabetes, but at the Digestive Disease Week. And what this is those same old biopsies, just in real time. You don't need to scope again, and we can do cell specific. You can look at the region of interest. We can show what's wrong, where it's wrong, and do something in real time. And so there is movement to move these types of tools to lab-based technologies at the point of care. And again, why would there be tryptase, which is a pain marker in your biopsies, if there's nothing wrong? So again, nursing science can do anything if you ask questions and you listen to your patients. What was I still coming back to? Oh, that pocket's still empty. Right? I don't have that Hemocult card. I've learned genetics. I'm still thinking back. I've now made it through. I'm a lab chief at NIH. I can do bigger protocols. I can have a global impact. We've got a protocol. We're ready to take some new technology, and where are people dying the most, and what is the biggest unmet need, and how can we know what to do in real time in rural health or at home in your bathroom, right? On an airplane, in outer space. And it's, diarrhea. Diarrhea kills. right? This is a global problem. We're going into a famine right now in our world. We don't have clean water, right? This is a problem. How can you make it more sort of US centric? You could think about detection of pathogens with rejection, with cancer medications. You could think about it from organ transplant, inflammatory bowel. You could think about resistant infections in hospital systems. But I had learned a lot about genetics. And what I learned was it's just a bunch of A's and U's and T's and G's. I mean, they're just in different orders. And I'd had all this data background, which is just a bunch of zeros and ones, and you organize them a different way. How is it any different? So I thought, can't we just find what's unique about a bug and then figure it out? And we did. So we created, this is pre-COVID, remind you. We basically just figured out what was unique in different pathogens that we'd see, and we figured out a way to test and use gold nanoparticles, tiny little bit, and biotinylated the tail, and this is all in a patent that's publicly available. You can look for it. We figured out how to do it just for one pathogen at the point of care, where you didn't need that dropper, right? You didn't need to know if you were colorblind. It's just a drop on it could be water, it could be stool, it could be, sorry, we all just ate, but vomit, et cetera, right? And then we could multiplex it. So if you were in an endemic area, you could pick different bugs, right? And for me, this was just nucleic acids, right? It's just different combinations. And so we were able to do this. We were able to get what's called SBIR, which is Small Business Research Initiatives. We went to phase 1, phase 2. We won Shark Tanks, all sorts of things back in 2018, 2019, just before the pandemic. And then for me, what I noticed was the level of detection. You had to have enough of the nucleic acids for it to light up. You guys have all had your COVID test. You know it. You're like, oh, is it still popping hot or not, right? Anyway, this is a different kind of technology, but we developed this little paper base because I didn't want to use plastic. So a little paper base kind of origami, and I've got one in my wallet if you want to touch it. Hasn't had anything on it, don't worry. And basically it folds up, and then you put it on top of just a little test tube. And this was glass-based. And this is from veterinary medicine, actually. It's called a float test. Oop, let me go back. See if I can make it run. And so we biotinylated little buoys so that within a substrate, it would float to the top. So say it's in your water supply, say it's in your food, et cetera. And then what would float to the top was highly concentrated pathogens. And then we could take our little origami thing and just put it on top, and it would wick, and then it would light up that way. And so it'd be really light, really cheap, less than $1, and direct to consumers. So this is being used by the DoD. It went on to, I-Corps funding and so forth. It's being used for sepsis and neonatal sepsis and around the world, more so from a military perspective for persons that are, say, in the field and don't have access because you don't need centrifugation or reagents and so forth to know that you have a pathogen present. So then what happened? COVID happened. And I'll sidestep a little bit. I left the NIH. I went to do implementation science. I got recruited to University of Connecticut because Fogarty wasn't turned off in 2019 before I left, but we couldn't be told yes. So I'd either rest on my laurels for the next 10 years till I could collect my pension, or I'd take a risk and leave. And so I left. And then COVID hit. And you're like, oh, wow. And so meanwhile, as everyone knows, who would remember in the lab space, if you weren't doing COVID-related work, your lab had to be morphed to a level 3 lab. I could still collaborate with level 3 labs. You can read some of the Nature papers we put out about detection of pathogens and so forth. I won't go into all of that now. During that time with Nihal out in Benet, a lab from NHLBI, which is the Heart, Lung, and Blood Institute. But meanwhile, Rockefeller still had patients, and they had a metabolic kitchen. And I always wanted to ask that question about sugar and high fructose, and all the corn lobbyers would have come after me if I did that as a federal worker. So we actually did this study. We did this study over COVID times. We looked at gut permeability. We taught the lab how to, the pharmacy to brew up our brain gut test solution. We used the GI pain pointer. We did microbiome. We did intra-abdominal fat. You name it. We could do it all, right? Because we could, right? It was Rockefeller, and no one could do anything, right, at that time. And guess what we found? So this is a randomized crossover design study, fructose versus sucrose, standardized metabolic kitchen, same amount of sucrose, same amount of physical activity, in-house, 2 weeks, absolutely nothing. We found absolutely nothing. I thought this would be the best pilot data for the next study that I could tell every kid in the world, do not do this, whatever. No, luckily it didn't make prime time, but it probably would have said something like, you can go on a cruise for 2 weeks and eat anything you want, do whatever you want, and you'll be just fine. So at any rate, lots of flaws, lots of stress and other things going on there. But it was a really great opportunity to work with amazing researchers that had similar questions. And they still have similar questions that we don't know the answer to. We don't know about lysozyme. Were you born that way? Did you use it up? We don't know how gut permeability changes across a lifespan, across a different disease state. We don't know how the drugs that you're giving will affect someone differently. And might their gut permeability tell you how much? Is it their size or is it their gastric accommodation size? Right? What if you change that? How does it affect? What will you give? What will you learn? What will your question be? So I'm back to home base again, right? How am I going to apply what I've learned? How can we take this data? So the people in the microbiome world look at data one way, people in the genetic world look at data another way, and the two shall not meet until now. So when I came to the University of Pennsylvania, they're like, oh, no, this is protected. This is our silo. Oh, this is our silo. And then I just had to find someone that was willing to look at O's and ones. It's all O's and ones. How can we make it all de-identified and look at the mechanisms? And sure enough, we were able to show that the microbiome, this is oral microbiome. We were tired of playing with stool. Sorry. I was going to say poop, but, and so from the oral microbiome, so just some cells in the mouth, we could basically tell what you need to eat and what you need to do differently, aside from telling you just don't be stressed, right? Because we can't change the world we're in right now, although we'd like to, but nurses have a role and have a question. So what are we doing now? We are working on abdominal pain still with engineering. Again, more of the simple science, me going back to, okay, what about the people who can't dial up on that GI pain pointer and they can't tell me how they feel, right? So again, starting in adults, because we have to, right? This is a belt that senses electrodermal activity and heart rate variability and bloat and so forth. And then we recapitulate this. This is funded from NIDDK, and year one, it's a 5-year grant, R01, which is a research grant. And year one was just developing the belt. So we had to do 12 different designs of belts, waterproof, this, that, the other. It's hot, cold, the seasons are crazy, and different sizes. So year one, we developed the belt. Year two, we had to recapitulate the symptoms to see it. You have to have pain to validate the belt, right? So we're just finishing year two, where we're validating the belt. And then years 3 through 5 will be a randomized controlled intervention that'll look at and have different interventions that we know work, but we don't know who works for what, when, and we're going to have the data to show indeed. Not only is there evidence, but the problem exists, right? And so thinking back to you, new grads, seasoned professionals, family members, we have a place in this world and we have a purpose and we have to have value, but it's value to not just what our patients tell us, but what our value is as nursing and what we give and our questions that we rise to the surface from our patients, from our communities, from the people that we serve. And so I just wanted to thank you for letting me be here, tell you about the Henderson Lab. We're always welcoming new thoughts. We like to bring people together that think differently. We're looking for disruptive technologies, not the next more expensive, better mousetrap or phone or whatever. We're looking for simple, really simple ways to help people help themselves. And so lots of references, but lots of acknowledgments and many thanks to Dean Annette Osaki. Nursing science matters. You will have many questions in the future. Build your network, your community of scholars. That anchor, it's people, and we come to different places, but we stay connected. And so I just welcome you all to contact me if you have any other questions. Are there questions? Yes, please. Yeah, Mike. Hold on. Yeah.
Thank you so much. That was just a really exciting. Your mind is amazing to witness. My question is why H. pylori doesn't show up in any of the bacterial,
Oh, H. pylori?
Yeah.
So we screened for H. pylori. And so for participants that screened positive for H. pylori, that actually there were enough individuals that screened positive that gave me the evidence to start what's called a natural history protocol, where we could actually treat in the nursing institute. Imagine treating patients and then follow them separately. So it's a separate protocol. Yep. Great question. And, there already is a little cassette that you can put a biopsy in when you're in the endoscopy that'll light up in real time for H. pylori. Yeah. So it was didn't feel like we needed that one. Mm-hmm.
Yeah, great. I want to thank you. And, of course, I'm sort of interested too in, sometimes I feel like in terms of our prescribing practices with antibiotics and how all of that might play and change some of what you were telling us in your story here today.
So the question is, how would my prescribing practices with antibiotics change?
Yeah, I'm just thinking sometimes, people are prescribing what I'm going to call this broad spectrum antibiotics. They just go after it with a nuclear bomb approach.
Absolutely.
Rather than we don't sometimes spend a specificity, right?
Specificity. Indeed.
Yeah. So I'm just wondering, do you see any of this reflected in any of the things that you've been looking at, thinking about gut health and C. diff and all these other issues?
Absolutely. Absolutely. And again, there are other kind of players in the field that are looking at, how different antibiotics. There's actually a student in the lab that specifically asked this question. Her name is Stephanie Prescott. She did a postdoc. I encourage you to look up her work. She purposefully used different cocktails of antibiotics that would kind of recapitulate, for example, if you'd for pregnant women before they give birth that test positive. So about 50% of women, right, either get a C-section or test positive. What is it for? Say it for me. Group B strep. Is that right? And so we see it. There's a 2-week washout. Typically, people come back to their baseline. And animal models typically come back to their baseline after 2 weeks unless you're immunocompromised. What's interesting is how do you get to that baseline, right? And that's from my health, sort of the health promotion perspective. And we find in early life that really the first year of life really dictates your microbiome for life. So, right, again, this is my pediatric, so I always go back to it's early life antibiotics within that first year that really prime that gut priming of the lifetime for response outside of, immunocompromised and so forth. So typically antibiotics, you'll bounce back. You may get a resistant bug, but if you're otherwise healthy, you typically will bounce back. But what you're bouncing back to may not be normal. It's your normal, but it may be likely to be disease producing.
Well, so that sort of raises the question about, so if you are treated with broad spectrum antibiotics and you're, changing the gut along the way, then how do you kind of try to get it recalibrated? So is there a role for probiotics?
Oh, yeah, absolutely. And there are certain, I'm trying not to name drop medications or names of things or names of companies, but there is a particular medication that helps 50% of individuals that have diarrheal predominant irritable bowel. That is a combination of antibiotics that we typically use a probiotic with. Again, my mind goes more to what is kind of the broader use and what people don't realize. For example, PPIs, what is that? Proton pump inhibitors. You guys are graduating, right? Or you're already out there. Have bacteriostatic properties. They're not meant to be used long-term, and people are using them over the counter long-term. So there are other kind of bigger, in my mind, kind of bigger questions where not to jump to treatment and to think about other holistic practices before we go to something that can actually change that milieu. So I'm not really answering your question. I think resistance is another huge, huge problem and issue on surfaces, even in hospital settings. It's interesting that it's now global, really. Yeah. But again, not my absolute area of expertise.
Yeah, thank you. My last question,
Please.
not that I should be asking all the questions here. I'm going to give this to Carly in a minute. But, you had to make a lot of pivots.
Oh, yes. That's
learn a lot of things. So for our students and people that are like thinking about maybe being scientists, maybe you should share a little bit about, what is the challenge that you faced in trying to make all these pivots when most people think, well, I'm just a clinician.
Absolutely. So I had the pleasure of meeting Abigail, a pediatric nurse, 80 years old, on the plane on the way here. And I was like, okay, the master of the universe brought us together on this airplane. And I was like, oh, can you tell me like what's going on? She's like, oh yeah, the children's hospital, this, that, the other. Turned out she was a palliative care pediatric nurse. Something I was like, oh my golly, how did you do that for so long? What did you do? And what advice would you give? I wanted to know what's happening in Buffalo and how can I like say something meaningful, et cetera, et cetera. She's like, you just need to tell them to lead with their heart. Right. And find people you know and trust along the way, that you can call up and can hold your hand as you pivot. And I think her thoughts and her comments were so telling. And I think in this uncertain time, of course I got to say, go with your gut, right? To that gut sensing. But, it's important. You've got to take some risks. But if you know it's the right thing to do, it's okay.
Other questions? Who else needs the microphone?
Hi. Oh, hello. I'm a pediatric nurse practitioner and midwife here at UB, and I loved your talk, and particularly because yesterday I met with a dentist at the Dental Institute at University of Rochester, and she was literally talking about the microbiome and the changes, and she was talking about children and infants and infant formula versus breastfeeding, and she said the microbiome basically is set by age 2. And we've done these studies that, in adults where you can make it move this way, but eventually it'll sort of just reset that way. And it just got my brain going. And of course, then today I'm listening to you thinking, well, what do we do about this? How do we actually look at this in a, prenatal, perinatal infant formula, where in the formula in Europe is totally different than it is here, because it has only the lactose there, but here it has sucrose and corn syrup and all kinds of good stuff to make kids think they're drinking Coca-Cola, I think. But anyway, I'm curious what you found for that. How do you set that microbiome earlier, so that it's on that right trajectory?
It's a beautiful question. And again, not my ideas, right? So Xiaomei Kong actually has done some of this work and is currently doing it, where she's looking at, again, it's that combination of what you eat and the stress mechanism, right? Where she's combining kangaroo care or touch, early in life with the birthing person, right? And the parents, and then breastfeeding, right? And so even breast milk may not always be good. There's donor milk as well. There are studies with donor milk. It may not also be the best thing either. And not all persons can breastfeed or chest feed, right? So it's sort of like, if we know that it's the combination of the two things that are affecting, like if we can at least do one, right? So if you can't give, breast milk from the birthing person, then, at least if we can have that positive anti-stress environment and so forth. And so I think that, our science overlaps, but I think that there's a lot more coming. And I think that now more than ever with the interest, and again, I'm the positivist. Like I'm always going, okay, there's got to be a positive way to make this pivot in this current environment. And there's a pivot to nutrition, right? And so how can we make that change now? How can we advocate for that? And so I'm just working in circles, international and otherwise, to be a part of that and to actually partner with industry and to, as well as, venture capital and so forth, to be thoughtful about our food supply, right? And so it's not just, right? And we've got to sleep, right? If you don't want to be stressed, we've got to have this holistic perspective. But I hope I answered your question. Yep.
Other questions?
Hi, Dr. Henderson. Fascinating presentation. I'm a first-year PhD nursing student. Yay, right? With someone who has celiac, and I've had it for a decade now. You got me thinking with talking about increased stress plus our diet and the role that carbohydrates, since butyrate is like one of the common things. So you got me thinking here. I'm interested in studying toxic exposures, predominantly, maybe postdoc, the role that pesticides have played, because if you think about grain and all the grains, right, especially wheat and corn, if you've ever been interested or are down the road to think about like carbohydrates are just, if they're not organic, they're full of pesticides and what role that plays in the gut. Thank you.
Yes, thank you. And congratulations. That was the other pitch I wanted to make to everyone finishing. I think I said it in the lab yesterday. Stay in school, stay in just a little bit, stay in school, keep your questions coming. And the other part of that is the genetics, right? Just because something is organic doesn't mean it's not genetically modified. So there's always going to be another question, and just keep them in, keep them in that pocket and be ready to take them out and keep asking questions and learn and ask it so that other people can be critical too in a positive way for how you can not only learn how to do the research, but then in the future, your dream question that you can have all those different variables somehow. Yeah. Question in the back. Thank you all so much.
Hi, I'm Sophia. I am faculty
Hi, Sophia.
DNP student, psych. But what was interesting is that you mentioned the PPI.
You talk right into the mic.
Oh, can you guys hear me?
Yes, better. Thank you.
So the PPIs, it's chronically used, and like you said, they're used for long term. So when someone chronically uses PPIs, is there a way that they can go back to an original, like, healthier gut health? And the second question I have is, our providers, other nurse practitioners, gastroenterologists, are they embracing a different treatment modality instead of the go-to of a PPI, since we know that it's not good for us in a long-term setting or care?
Amazing questions. Again, I come from the pediatric perspective, so I can't speak for the adult world, right? But in the pediatric perspective, the goal is to, if biopsy is indicated, to do it first before there's a bacteriostatic or other properties introduced, so that you can find, for example, H. pylori or otherwise, which you may miss with PPIs. The other thing is PPIs tend to stop working for people over time, and you typically have to cycle them. And so what we try to do, at least in the pediatric emerging adult world, is when the cycle is timed, then we try to kind of do a washout. And so we've published some treatment algorithms, even back to like a full elemental diet and kind of repriming the gut entirely, sort of a clean out, restart, reintroduce one food at a time, and then look at triggers, because there must be a reason, right? There must be evidence that there's a problem exists, and not just treating the symptoms, right? So I think that's kind of where the science is headed. Yes, please.
Hi, I'm Daniel Smith, faculty and a nurse practitioner. And just for those of us in the room who do more social determinants of health, so I think of my clinical practice at the City Mission with the unhoused population. How does your work potentially benefit those of us in the SDOH space, and what connections can you make? Because I just hear gut microbiome, and I think this is a technology that my patients will never have access to.
Right, because they need access to food, they need access to clean water, right? To have a safe place that they call home, right? So what isn't published, and research that we did do in-house, and what we could best phenotype individuals in our protocols, because lots of people would come to the NIH because we could pay them to be in a study, right? And so persons from all over, right? Or from nowhere entirely. And what was most interesting to us was that we could phenotype their microbiome by their water supply. So we could know best their healthful microbiome just by what they drank. And people can live all sorts of different areas, right? And there's some value to actually not clean water, right? In terms of health and avoiding disease too. So I think that, again, it's not my area per se. We do try to think about technologies that are useful to all, where they can kind of morph it however they'd like it. But come on, what if you don't have electricity, right? Like, how are you going to use the GI pain pointer, right? How is that useful? But being able to, like, say, here's some strips, and you can see if you're dehydrated or not. Or here's, right? I mean, I'm sure you're doing this, right? When it's really hot or otherwise. Here's how you could know if you have something so that you don't share it with your partner, right? If it's, for example, a virus, which is also part of the patents. And just simple, where you don't have to go to a clinic. You don't have to be identified to be able to take care of yourself and know what's wrong with you. And you can have that information and kept to yourself, I think, is another thing that's really valuable from a social determinants perspective, where you can still share the information, but it's not like someone's coming in or taking control of your health. Is that helpful at all?
It is, and it makes me think to, so before I was at UB, I was at Villanova and Philly, right into
Mhm.
Kensington, and we had a Shigella outbreak.
Oh,
And
sure.
so it was like we were treating people in the street for presumptive Shigella, but again, that's just because people were homeless and living on the street. So we just, if it walks like a duck, it's a duck, right? So something like that
Yeah. Yeah.
have been very useful at the time.
Yeah.
Thank
Great
you.
question. Great question. Thank you. All right. Well, thank you all. I'm between you and your posters and lunch. So thank you. Take care.